Chapter 3 will take us to the next level of organization, which will be the cellular level organization. This will be the last chapter that should be relatively familiar to most of you from either high school biology or from biology 1 and 2 based on the college level. In this chapter, we'll talk about the different parts of the animal cell. We'll talk about the importance of the plasma membrane, how it's formed, why it's formed the way it is, and what kind of structures can pass through it with and without help. We'll talk about the components of the cytoplasm, the fluid that's found within the cell or the intracellular fluid. And we'll talk about the different mechanisms of metabolism that occurs into there as well. We'll both compare and contrast both mitosis and meiosis. This will be very important. We go through the lab activities coming up this week. And finally, we'll talk about how aging will affect the cell. So the cell can be subdivided into three parts. We have the plasma membrane, which basically separates the extracellular fluid to the intracellular fluid. Sometimes you'll see different gradients of different solutes or possibly different voltage from intracellular to extracellular fluids. This is very important, of course, for conducting any kind of nerve transmission along the pathway of a nerve. The second part will be the cytoplasm, which includes the cytosol, the actual fluid, and of course all the organelles found within the plasma membrane. Finally, we'll talk about the nucleus, where of course we find the chromosomes and also the genes. These are templates for building proteins, as well as very important for cellular mitosis. We'll talk about a special structure found within the nucleus called the nucleolus, which is the centermost portion of the nucleus, the most condensed portion of the chromosomes and genes. Here we see a representation of a typical animal cell. This has all the structures that could possibly be found within an animal cell. Unfortunately, we do not have any cells in our body that will have all of these structures together. In fact, the only cell that has a flagellum, which is that whip-like protrusion on the upper portion of the slide here, is only found in male sperm. Other than that, the other structures can be found more differentiated depending on what kind of cell that we're talking about. But you can see clearly there's the nucleus, which is going to be enveloped by the nuclear envelope, which we'll go through in more detail later on. Nuclear pores for where mRNA is going to pass through to be translated into amino acid form. You have the cytoplasm, which is going to include everything except for the nucleus. You're going to find both a smooth and rough endoplasmic reticulum. The rough shows the ribosomes that will be attached to the membrane, whereas the smooth endoplasmic reticulum will not. We'll see in the orange, almost dead center of the slide here, see the Golgi complex. Used to be known as the Golgi apparatus or Golgi body. You'll see kidney-shaped beamed structures found all throughout the cell here. They're mitochondria. These are the powerhouses of the cell. You'll see small projections off of the cell membrane, including the microvilli, or possibly the cilium, which we'll talk about structures with those later on, as well as microtubules. And of course, you'll see different things floating within the cytoplasm, which include the secretory vesicles, which will fuse with the plasma membrane to expel any kind of product. Lysosomes, they're very important for breaking down product, as well as a few other structures. Also see microfilaments and microtubules, which give structure to the actual cell. Some of these will be contractile, which we'll find in muscle cells in particular that will produce motion. The first structure we'll identify is the plasma membrane. It is a lipid bilayer composed of phospholipids. If you imagine a bunch of balloons that are pretty much floating all throughout, it's a very fluid mosaic model. So very simply, these little balloons or the phospholipids will move freely on one side, and very, very rarely will they flip to the other side. In other words, you have dedicated phospholipids on the extracellular side and then dedicated phospholipids on the intracellular side. Notice scattered all throughout, we have different types of proteins. They'll either be on either side of the plasma membrane, known as the peripheral proteins. So you see one peripheral protein is going to be on the extracellular area, and on the flip side, you'll see the peripheral proteins found in the intracellular side. Now, ones that will span both layers of the plasma membrane are be known as integral or transmembrane proteins. You'll find these will be channel proteins. They'll open up pores. Some are selectively permeable. Some open automatically just through random chance, and some will open through different changes in voltage going across the actual cell, including nerve cells and also muscle cells. We also have glycoproteins, which is exactly what it sounds like. It's a combination of protein and carbohydrate together. Red blood cells will be tagged with these to identify different types of blood types. And of course, scattered within the phospholipid bilayer, you're going to find little pieces of cholesterol, which is very important for keeping the membrane together. Too little cholesterol and it'll break apart. Too much cholesterol and actually you start to lose fluidity of the lipid bilayer and once you start to solidify. Last structure you see there is the glycolipid, which is of course a combination of lipid and carbohydrate together. These will help make the plasma membrane less sticky so they can slide upon one another. Very important for white blood cells in particular that will travel between other cells to get to wherever the target may be, whether it be to reduce antibodies or physically attack a foreign substance or a foreign cell. So again, here are the two types of membrane proteins that we'll find. We'll find the integral or the transmembrane proteins, which again will channel all through both layers of the phospholipid bilayer. Otherwise, we'll find the peripheral proteins used for identification or for possibly for metabolism on the extracellular fluid side. You'll also have other peripheral proteins found in the intracellular fluid. This might actually aid in the actual structure of the cell. So, for example, integumentary skin cells hold a very particular structure and get anchored together. These different types of peripheral proteins, which we'll talk about later, help keep these pieces together. Other types of peripheral proteins would include your actin and myosin that you'll find within muscle cells that provide the contractility to allow proper motion. So the myriad of different types of membrane proteins can have numerous functions. In this case, we're going to show 6 here, which would make an excellent type of test question. Very simply, they can be used as an ion channel. Very important to allow proper ion flow, either against or with the particular gradients, which we'll get into later on. So ions would include your positively and negatively charged ions, which would include potassium, calcium, sodium, chloride in particular. Some of these will open randomly, and some of these will work against gradients, so it'll need energy, in this case ATP. Again, they'll take care of mostly the smaller molecules that'll pass through these particular pores. Anything that's larger than that will need what's known as a carrier protein, which is also an integral protein. So larger molecules such as glucose will have to pass through the carrier. Very simply, the protein will accept that particular molecule, it'll change composition and actually push it through the plasma membrane. Again, this can be done either with or against gradients. Next one up is a receptor type. We'll see this a lot, especially with neurotransmitters and dealing with neurons. So going from different synapses, let's say different types of neurotransmitters like acetylcholine or norepinephrine. Basically, the ligand will bind with the active site at the receptor and change the conformation of a particular cell. It shows kidney cells in particular will change the permeability of the plasma membranes to allow proper flow as far as fluids go. You also have enzymes, of course, that will take different types of substrates and change them into different types of products. So in this case, turning milk sugar into monosaccharides instead of the disaccharide lactose, it'll split them up appropriately into galactose and glucose. You have linker proteins, very important for the basement membrane found in the integumentary system to hold them together. Also very important for the epithelial lining of your stomach cell to make sure no hydrochloric acid will sift through and destroy the underlying basement membrane. And finally, you'll find cell identity markers, which are known as glycoproteins. You'll find these especially on red blood cells as well as other cells throughout the body, so your immune system can be able to identify which one's considered to be foreign and which one's considered to be friendly, so to speak. The fluid mosaic model, the membrane has fluidity, which basically means that different lipids and the majority of the membrane proteins will move very easily through its own section of the bilayer. Most of the time, the proteins will not switch to, say, the extracellular fluid side to the intracellular fluid side, but occasionally the actual phospholipids may flip from time to time, but that is extremely rare. As I mentioned before, cholesterol serves a major purpose with trying to keep the membrane fluid. You have too much cholesterol and it will almost solidify. Not enough cholesterol found within the plasma membrane and it will basically dissipate and the cell will essentially lyse. Another aspect of the plasma membrane is its permeability. So very simply, depending on the structure, certain things can pass through the lipid bilayer very easily. Some will pass through certain channels, or some may need some extra help or be engulfed by the cell itself. So in this case, small, nonpolar, uncharged molecules, which includes water, oxygen, carbon dioxide, this will slide through the lipid bilayer pretty easily. Those that have any kind of charge, say the potassium or chloride or sodium, these have to actually pass through transmembrane proteins or channels or transporters to basically bypass the permeability of the membrane. Anything larger than these particular types of molecules, in this case glucose, some amino acids, some proteins, these are the macromolecules. These will only pass through the plasma membrane by vesicular transport, which basically will fuse with the plasma membrane and either import macromolecules into the cell, or on the flip side, export it out into extracellular fluid. The presence of the plasma membrane and also the selective permeability of the membrane allows us to have different types of gradients. So very simply, a concentration gradient is the difference of a concentration of one particular chemical, both in the intracellular fluid versus the extracellular fluid. So that could be either carbon dioxide versus oxygen, as you see here, sodium versus chloride. It could be glucose in the blood coming into the cell, or concentrated in the cell going out into the blood. The other type of gradient we see is with ions. So if there's a difference of ions between the intracellular and extracellular layers of the plasma membrane, that's called the electrical gradient. Very important for nerve cells and also for muscle cells as they travel along the plasma membrane. Together, this was known as the electrochemical gradient, which we'll get into a little more so when we get into different types of tissues. We see transport across the plasma membrane in one of two ways. We see either passive processes, which will occur when you're taking something from a higher concentration to a lower concentration, or an active process where you're going against the concentration gradient, where you're going from a lower concentration and trying to drive things into a higher concentration. So very simply, the passive processes will include simple diffusion, where very simply the small nonpolar, non-charged molecules will simply pass through the plasma membrane. This will include your gases, including oxygen and carbon dioxide, as well as water. Osmosis is the simple diffusion of water itself. We all see facilitated diffusion, where very simply we have larger macromolecules, including glucose, amino acids, and other types of macros, will pass through the plasma membrane either through what's known as a channel-mediated diffusion or a carrier-mediated diffusion, which we'll get into in just a few moments. On the flip side, if you're going against the gradient, this requires energy, or requires ATP. So we have active transport, which will constantly pump, whether it be ions or macromolecules, against the concentration gradient. The most common most people know is the sodium-potassium pump found in nerve cells. You also have vesicular transport, where very simply you'll have an engulfing of certain macromolecules, or in some cases phagocytosis, where you're trying to ingest foreign substances, or penocytosis, or cell drinking, where you're forming vesicles to draw certain types of products in to basically break down for the cell to use for other means. There's a nice little animation that goes through everything we just talked about here. All different types of transport across the plasma membrane, including the passive and active processes. So next, let's go a little more in depth in the passive processes. First type of passive processes we're going to look at is simple diffusion, and there's going to be certain factors that will influence how quickly diffusion can occur. Number one, the steepness of the concentration gradient. Naturally, the greater the steepness, or greater change between the initial concentration on one end of the plasma membrane versus the other, can speed things along. So the more steep it is, the faster the process of diffusion will occur. Higher temperatures will cause more molecular movement, and therefore cause greater diffusion at a greater rate. The larger the mass of the diffusion substance is, the faster it will diffuse into equilibrium. The greater the surface area of the plasma membrane will also speed up the diffusion process. But if the diffusion distance is great, that will actually slow down the actual diffusion. So the smaller distance that it needs to occur to reach an equilibrium, the faster diffusion can occur. Now, when a molecule is either too large or has too much of a charge, we rely on facilitated diffusion to transport these particular macromolecules from extracellular fluid into the cell. Now, this can occur in one of two ways. It either go through a channel-mediated facilitated diffusion, which you use for most of your ions like potassium, chloride, sodium, and so forth. Very simply, this will pass through channels which will be randomly opened or closed, basically going from higher concentration to lower concentration, or the particular larger molecules, which might include amino acids, or may include glucose or other types of lipids and such. These will pass through carrier-mediated facilitated diffusion. Very simply, it'll bind with the transmembrane protein. The transmembrane protein will basically change its configuration and basically dump that particular molecule into the cell. Now, of course, the rate of diffusion for either one of these processes is entirely dependent on how many actual channels you have available. So there's only so many in the plasma membrane, then diffusion will only the rate will only be as fast as how many actual channels you may have in that particular area. So here we're seeing an example of the channel-mediated facilitated fusion. We see potassium that's going to flow out of the cell once the channel protein is open. If the K is closed, naturally the potassium is not able to leave the cell. Here we see carrier-mediated facilitated fusion, where you see glucose molecules will bind with the glucose transporter. So essentially, we're taking this from the blood and going, say, into a liver cell. It's going to change configuration, as you see in step 2 and 3, and dumps glucose into the cytosol. So here you see the comparison. So very simply, all of these are going through from a higher concentration to lower concentration. So for example, simple diffusion, you'll see water, you'll see oxygen, carbon dioxide passing right through the bilayer without much of an issue. If they're charged ions, in particular your calcium, potassium, sodium, and so forth, these go through channel-mediated facilitated fusion. As long as the channel port is open, it'll pass through the channel from higher to lowest concentration. This is very important, in particular, once we have a nerve that has fired off or had an action potential, to try and bounce everything back out from the electrochemical gradient. These random channels will open up to allow proper ions to bounce out from the extracellular fluid to the cytosol. And finally, you see on the far right there the carrier-mediated facilitated fusion. So in this case, glucose will bind with the channel with the transmembrane protein. It will change configuration and then push glucose into the cytosol. As I mentioned before, osmosis is simply the diffusion of water. And one thing to always remember with osmosis is water will always follow the higher concentration of salts. So in this case, very simply, you're going to see at the start of the experiment, you have a selectively permeable membrane, and you have left and right arm there. So at the start of the experiment, you see obviously that the solid molecules on the right can't pass through the selectively permeable membrane. Then what happens through osmosis and hydrostatic pressure, more of the fluid will pass to the right side to balance out, because naturally there's more salts on the right arm there because of the smaller and larger molecules there. When you apply osmotic pressure, it'll actually equal out everything back to the starting conditions, but once that pressure is taken away, it'll go back to equilibrium state that you see at letter B on the diagram here. So very simply, it's all dependent on the permeability of the membrane and what kind of solids can pass through easily or which ones cannot pass very easily. But no matter what, again, the water will follow the concentration of salts. Higher concentration of salt, the more water will flow. We'll see how this is very important, in particular when we deal with red blood cells and other cells found throughout the body. Now here we use the properties of osmosis to talk about tonicity. So very simply, we're looking at the relationship between the solution and the cells we found within the solution. So very simply, if the equal amount of water flows in and out of the cell equally, the cell is known to be in what's called an isotonic solution. So again, equal number of salts found in the fluid itself and also in the red blood cell, as you see here on letter A. We have a red blood cell in a hypotonic solution, where very simply there's a large concentration of salts found in the red blood cell and less so in the solution itself. What happens is through osmosis, the water will rush in, and eventually what ends up happening is the pressure, osmotic pressure, is insustainable to the plasma membrane. So the plasma membrane just simply breaks apart, and you have what's called hemolysis, where the red blood cell just ruptures. And on the flip side, if you have a red blood cell in what's known as a hypertonic solution, whereas high concentration of salts in the hypertonic solution, what happens is water rushes out of the cell, because as we said before, the water always follows the higher concentration of salts, and then the red blood cell will crenate or start to curdle together. And again, that's also will destroy the cell just easily as causing the hemolysis on the red blood cell in letter B. Next, let's talk about the active processes. So very simply, these require energy, very simply because we're going against the concentration gradient. So to move things against the concentration gradient, we use primary active transport. So very simply, we have here a sodium-potassium pump. We find most commonly in nerve cells. Very simply, after action potential has been reached, the cell tries to get back to what's called a resting membrane potential. So very simply, the sodium-potassium pump, or the sodium-potassium ATPase, will basically take 3 sodium molecules and then very simply ATP, ACE will break down ADP and phosphate, and use that energy will change the configuration of the actual protein and expel the sodium. The phosphate will bind with the protein, and very simply when it accepts two potassiums, it'll release the phosphate and therefore change configuration again and try and import in two potassium molecules. So again, having the ADP and also the P freestanding the cytosol, usually ATP synthase will actually reform ATP at that point, and then basically the cycle can be replenished over and over again. Now we also have what's called secondary active transport, which basically is going to use the concentration gradient of one particular substance, in this case sodium, where it's highly concentrated in extracellular fluid and it's fighting to get into the cell, into the cytosol. We're using that gradient to help us out. Even though this still requires ATP, it's being used indirectly. So very simply, we have two types of secondary active transport. You have what's called an antiport, where it's going to draw in one molecule, in this case sodium, and expel another one going in the opposite direction. So in this case, for the antiport, it's going to draw in the sodium from higher concentration to lower concentration, but it's going to expel either a calcium ion, where it's going to eject it into the extracellular fluid against this gradient, or the hydrogen ion that you see in the second part of the antiport section there. Or you have what's called a symporter, where very simply we're using the sodium gradient to aid us in also drawing in a secondary molecule, in this case either glucose or amino acid. Again, still requiring ATP, but because it's being used indirectly, this is known as secondary active transport. So again, in this case, we're going to use one molecule that's going to flow with a gradient to either expel other molecules against the gradient into the extracellular fluid, in the case of an antiporter, or in the case of a symporter, we're trying to draw in the same direction, try to draw in another molecule, in this case glucose or amino acid, along with the sodium ions. Now another way we draw certain substances into the cells when they're either too large or they cannot fit through the actual membrane proteins, we do different types of active transport. In this case, we're looking at what's called receptor-mediated endocytosis. In this case, we have a low-density lipoprotein, which is going to bind with receptors, and what will happen is that will actually cause the area around the plasma membrane to start to invaginate a bit. You'll have what's called a clathrin-coated pit. It'll start to close itself off and then basically fuse to form a vesicle. And what you'll see here on the step 2, you'll see the clathrin coat, the vesicle there. The clathrin will actually start to break apart and either be recycled back onto the plasma membrane or in different parts of the cytosol. Then it becomes uncoated, so you have an uncoated vesicle. It'll fuse with an endosome, which will very simply recycle the rest of the receptors and also break down the individual lipoproteins. That will later fuse with a lysosome, and very simply the lysosome will break down into smaller products for the cell to use for a different purpose. So again, this is known as receptor-mediated endocytosis, so it's drawing in this case, lipoproteins into the cell to be broken down into smaller products. At the same time, you're going to be recycling both the clathrin coat as well as the receptors to be fused either back of the plasma membrane or simply broken down and recycled to another product that the cell can use. Now next we see phagocytosis, which has some similarity to receptor-mediated endocytosis, except there's no clathrin pit. Instead, we have the receptors, they'll bind with the microbe, and in this case a white blood cell will protract pseudopods, or little arm-like projections, which will envelop the microbe and the receptors together into what's called a phagosome. The phagosome will fuse with a lysosome, which will include the digestive enzymes. It'll break down the microbe, and it'll be left with the residual body either to use wherever products there may be, either within the cell or possibly expel it back into the blood area. And the last type of active transport that we see in vesicles is known as bulk phase endocytosis, otherwise known as penocytosis. This is what's called cell drinking. So with this, there's no receptors involved. So what happens is the formation of the vesicle will actually draw in the fluid and the solutes together. You see this oftentimes in kidneys and also intestinal cells. So you form a vesicle with the fluid and dissolve solutes in the vesicle. It'll fuse with the lysosome. The lysosome will further break down the solutes into smaller macromolecules to be used elsewhere throughout the cell. Now, oftentimes we see with active transport, you can see two other types of processes known as exocytosis or transcytosis. So exocytosis very simply is you have a circulatory vesicle with its constituents who actually fuse the plasma membrane on its way out and basically dump the contents into the extracellular fluid. You see this in particular with hormones, in particular with neurotransmitters, which we'll get into when we get into the nerve cell chapters. Occasionally in the epithelial cells, or you'll see this in gastrointestinal tracts, you'll get transcytosis, where it's a combination of endocytosis, where you're drawing something in on one side of the cell, transport across the cell, and then basically expelled out through exocytosis through either directly across or a different portion of the plasma membrane. Here's a quick little comparison of all the transport types for you to read through. Nice little cheat sheet for you. So it goes through all the passive processes, including diffusion, the two types, simple and facilitated, and of course osmosis is only the diffusion of water, not other molecules. And just be sure to understand how osmosis works, in particular when dealing with red blood cells. Active processes will include the active transports, which include primary versus secondary, as well as transport via vesicle, whether it be endocytosis, which includes receptor-mediated endocytosis, phagocytosis, or endocytosis bulk phase, and no difference between exocytosis and transcytosis. So exocytosis, again, is the expulsion with the fusion of the vesicles with the plasma membrane, and transcytosis, very simply, is the combination of endocytosis drawing in and exocytosis pushing outward on a different part of the plasma membrane. So again, when we talk about the cytoplasm, we talk about the plasma membrane. So the cytoplasm is going to include everything within the plasma membrane except for the nucleus. So the cytoplasm will include all the organelles except for the nucleus, as well as the cytosol. So again, the cytosol is known as the intracellular fluid. The organelles are going to be all the other specialized structures. They're going to have different types of shapes and different types of functions, which we'll go over in just a moment. Now, if you're going through your book, the table you're going to see here in this slide, next slide, is actually towards the end of this section, but I feel like this works better if I put it in front of all the individual organelles that we're going to talk about, because at least gives a nice little cheat sheet, at least kind of jar your memory back from high school biology about different parts of the cell itself. So we already talked about the plasma membrane, how it's the fluid mosaic model, how we find membranes and glycoproteins, so on and so forth, in and around the edges of the plasma membrane. It talks about different functions, how it maintains the shape of the cell, how it can act as transporters, receptors, enzymes, cell identity markers, and so on and so forth. We talked briefly about the cytoplasm, basically the area that's going to be between the plasma membrane and the nucleus itself, is where all these intracellular activities will occur, except within the nucleus. The cytosol is going to be the fluid or intracellular fluid, which includes water, solutes, lipid droplets, glycogen granules, and so on and so forth. Also within the cytosol, which you'll see in a couple slides, cytoskeleton, which is made of 3 different types of filaments: microfilaments, intermediate filaments, and microtubules. This helps give shape and stability to the individual cell. Some organelles you'll see will be the centrosome, which includes the centrioles, which will very important for developing the mitotic spindle for cell division. We have cilia and flagella, which we'll look at a little more closely in a couple slides, where cilia will be used to move certain things over the cell surface, and flagella is to move the entire cell. But again, flagella is only seen in male sperm in the human body. Ribosomes come in 2 types. There's free-floating within the cytosol, and others will be attached to the rough endoplasmic reticulum. This is what creates or starts the protein synthesis. So endoplasmic reticulum you'll find adjacent to the nucleus. Attached to the nuclear envelope, it's going to contain the rough ER for protein synthesis, glycoprotein synthesis, and for packaging. You have the Golgi complex, so it has both a cis and a trans face. Cis faces nucleus, trans faces the plasma membrane. This takes the proteins and repackages them into different types of vesicles, usually for transport either out of the cell or to a different organelle. Lysosomes, these are your vesicles that have digestive enzymes to break down, whether it be old proteins or whether it be to break down microbes and so on and so forth. You have peroxisomes, which are oxidases, very specialized lysosome. Proteasomes, which are vesicles that include proteases to help break down proteins into amino acids. Mitochondrion, which you always hear about, are the powerhouse of the cell. This is where ATP production will occur for cellular respiration. Breakdown of ATP and development of ADP and phosphates together. And finally, we'll talk a little more about the nucleus in a different portion of the lecture. Next, we move on to the cytoskeleton, which is made up of 3 different types of structures. We have microfilaments, which are usually made up of actin and myosin, and that should ring a bell. Actin and myosin are the proteins, the contractile proteins found in muscle cells, but we find these in a lot of different cells help maintain proper shape. In particular, you see these in epithelial cells, you see this in muscle cells, and also in portion neurons as well. The microfilaments are very abundant. They tend to hold shape through the microvillus, which you see at the top cell here. This increases surface area. It's very important for absorption in particular. Next, you have intermediate filaments. This helps maintain the shape of the cell and also helps anchor one cell to the other. So this is very important for cells under a lot of mechanical stress. So the basement membrane of epithelial cells in particular, basically tight knit to not allow anything to pass through that shouldn't. And next you have the microtubules, which will be consistent with the centrosomes. Besides also offering structural integrity to the cell itself, the microtubules are very important for maintaining the proper position of organelles, and also, more importantly, also helps form the mitotic spindle for mitosis in cells. Now here's a closer look of the centrosome and the centrioles. If you notice, they're arranged in a very particular pattern. They're arranged in triplets of tubulin, which is the protein that makes up the microtubules. It's in a 9x3 arrangement. So basically, this is where the mitotic spindle will generate from. Normally, when the cell is in interphase, the centrosome is relatively inert. But once the mitotic division starts, you see the centrosomes will actually start to migrate at the poles, start developing the mitotic spindle, and basically pull the 2 halves of the cell apart to create 2 daughter cells. Now, cilia and flagella, very similar to centrosomes, will also have the same kind of triplet arrangement. If you notice here, the basal body, which attaches the cilium or the flagella to the plasma membrane, has the same scenario as the centrosomes. It's a 9x3 orientation. And then the actual cilium and flagellum, it goes into doublet microtubules here. So you have a central pair as well as 8 surrounding pairs around. This helps provide motion back and forth. So very simply, those doublet microtubules, they slide on one another back and forth in reciprocating fashion, and that creates the whip-like or even a propeller type of motion in either cilia or flagella. So you can see in letter B, you see cilia lining the trachea. The cilia is more important for helping move fluid along in a certain direction, whereas flagellum is usually a single process here, and it's usually used in propeller motion for movement of the cell itself. Of course, you see in letter C, it is a sperm cell with a very long flagellum there for a proper locomotion. Now, both the cilia and flagella will have 2 different types of motions for 2 different purposes. The cilia in particular is trying to move fluid in a certain direction. So very simply, it'll start out in a very elongated position, as you see in the power stroke in the number 1 spot there. Very simply, it'll whip itself in the direction that wants to move the fluid. It was known as a power stroke. If you go through 1, 2, 3, 4, you see cilia whipping through. And then very simply, it'll contract and actually start to elongate back to its original position in what's called a recovery stroke. So again, this usually works in quite a large number of cilia, all trying to push fluid in a certain direction. As far as flagellar motion goes, we see one of 2 different types of motions. We see either a whip-like flagellar motion, very similar to the movement of a fish's tail or a shark's tail, or you may see go in a circular fashion in a propeller, very similar to a helicopter, to try and drive, in this case, sperm in a very direct position. Ribosomes will go into a little more thoroughly later on in the chapter. Very simply, just be aware that ribosomes are made of both a large and small subunit. You find these either free-floating in the cytosol or they'll be attached to the rough endoplasmic reticulum. Here we have the endoplasmic reticulum, which comes in 2 types. You have both the rough and the smooth ER. The rough ER will have the ribosomes, which again will be continuous with the nuclear envelope of the nucleus. So it's a very important site for synthesis of proteins, glycoproteins, and phospholipids. So very simply, a lot of these will be recycled into different organelles or possibly processed by the Golgi apparatus, which we'll look at in a few moments. These can either be exported into the cell through the Golgi apparatus as well. Next, you have the smooth endoplasmic reticulum. This is more so for fat and lipid synthesis. It will not have any ribosomes attached to it. This is also very important. Some smooth ER can be highly specialized. We see the sarcoplasmic reticulum, which is a version of the smooth ER found in muscle cells, which holds the calcium stored to allow proper muscle contraction. Here we have the Golgi complex, otherwise known as the Golgi apparatus. This will accept transport vesicles from the rough ER at the cis face. It's repackaged in different vesicles with different products, and will exit the trans face through cingulatory vesicles, usually either to a different organelle or possibly out to the plasma membrane. Basically, the cingulatory vesicle will fuse and then basically dump its products out into the extracellular fluid, or in some cases, it may go out into the external environment. A closer look of how the cingulatory vesicles or transport vesicles coming off the rough endoplasmic reticulum will merge up with the Golgi complex. Very simply, a transport vesicle is pinched off from the rough endoplasmic reticulum. The transport vesicle will fuse with the cis face of the Golgi complex, and it'll move through the medial cisterna to be repackaged or to be repurposed. This is where you find glycoproteins, lipoproteins, and other types of enzymes will be formed within the Golgi complex. Some will be formed in transport vesicle to go to a different organelle or different part of the cell. Some will be packaged towards the plasma membrane to do a number of things. It may actually secrete its constituents out into extracellular fluid. In this case, the expulsion of insulin or neurotransmitters are done this way here. Occasionally, it may have trying to replenish the plasma membrane. So if you look at number 8 in particular, it may have transmembrane proteins attached to it, or it may have peripheral proteins attached to it to replenish those that are lost or damaged on the plasma membrane. Or it may actually package enzymes and create itself basically a lysosome to break down other kind of foreign objects in the cell or any kind of products that may be needed for the cell to function. Lysosomes are transport vesicles with highly digestive enzymes, and these can be further differentiated in different types of lysosomes depending on what they're trying to break down. Now, peroxisomes are very important. These are mainly used to oxidize organic substances, very similar in shape and description as lysosomes, but have a very particular purpose. Again, this is more for oxidation or breakdown of organic substances. Proteasomes are another type of lysosome, except these will help destroy either unneeded or damaged or even faulty proteins by chopping them down from long protein chains into the smaller portions such as amino acids. Next, we have mitochondria, otherwise known as the powerhouse of the cell. This is where ATP is broken down to provide energy. Usually you find these very close to the plasma membrane as well as other organelles, interspersed all throughout the cell, where it takes in diffuse oxygen and use that to break the bonds, the phosphate bonds, and ATP to generate energy. The mitochondria is structured with both an outer and an inner mitochondrial membrane. And you see the many infoldings inside there. You have the mitochondrial matrix, which is its own little intracellular fluid found within the mitochondria, and mitochondrial cristae, which is where the breaking of ATP to ADP and phosphate to break these bonds for energy will occur. The mitochondria also has its own ribosomes. These will also self-replicate. Mitochondria have their own DNA, and usually you inherit these from your mother, very simply because the sperm from the father has very few organelles in it because it has to stay relatively light in order to penetrate the oocyte and be able to travel and get to it. So all the mitochondria that we have ourselves comes from our mother. Now, one other feature not generally known about mitochondria is it's very important in the process of apoptosis, which is cell death. When the mitochondria is or detects certain levels of stress, whether it be free radicals, breakdown of other types of proteins, the mitochondria will start releasing certain types of digestive enzymes to basically destroy the cell. For some areas in the body, this is a normal process. Cells become damaged from either DNA destruction or any kind of DNA mistakes during replication. So the body will actually self-destruct this particular cell. So the mitochondria plays a very big part in that. What we'll come to is the nucleus, or the master control center of the cell. Is where you find DNA replication and DNA translation to form mRNA and to form proteins later on in the cytosol. So features of the nucleus, we see this is going to be a double layered membrane, otherwise known as the nuclear envelope. You'll find scattered throughout will be nuclear pores. This is where proteins that are used for nuclear type of reactions will be diffused into the nucleus. But otherwise it normally exports mRNA out to be translated. More dense portion of the nucleus is known as the nucleolus, which is a more densely packed area of chromatin. You can also see ribosomes found all throughout the outer edge of the nuclear envelope, the polyribosomes, as it is continuous with the rough endoplasmic reticulum. Within the nucleus we find the genes, which are basically chromosomes wrapped around as a DNA double helix, wrapped around histones or proteins to form a nucleosome. These are held together by linker DNA in a more condensed formation of chromatin. When we get into an active mode of mitosis, it will condense into chromatids, which will be 4 chromatids with 2 sister chromatids on either side of a centralized centromere. As we see here, the primary function of DNA is for gene expression. Very simply, the base codons will be matched up with the opposing groups for RNA. As messenger RNA, it will be transported out of the nuclear envelope through the nuclear pore out into the cytoplasm, where it will link up with a ribosome and have the corresponding anticodons of the protein to form an amino acid chain and to therefore later on form a protein. We achieve protein synthesis by way of transcription. This happens in the nucleus. It's a process by which the DNA is unzipped by a protein called RNA polymerase, and a messenger RNA is going to be opposed with based pairs from the DNA. Only on one side of the DNA strand, not both sides, will actually have the same type of protein, but only one side will correspond to the messenger RNA that's going to be sequenced for a certain type of protein. When we look at the base pairs, if you remember with DNA, you're going to have adenine, guanine, thymine, and cytosine. These will oppose the messenger RNA, so adenine will pair with guanine instead of thymine. Guanine is going to be particular for RNA only. Thymine will pair with adenine, guanine will pair with cytosine, and cytosine will pair with guanine. In this case here, you see the RNA polymerase will have the matching codon, which will correspond opposing the base triplet on the DNA. This will basically form a pre-messenger RNA. Basically will have a start codon or an intron, as well as exons that will be cut by small nuclear ribonuclear proteins to basically give a more functional mRNA. So non-functional introns will be snipped or snurped in this case to correspond with the small nuclear ribonuclear proteins to be ready to be transported out the nuclear pore into the cytosol, and eventually will pair up with a ribosome for translation. Once the mRNA nucleotide sequence exits the nuclear pore and goes into the cytosol, translation actually occurs not in the nucleus. This is actually incorrect on the slide here. Translation occurs in the cytoplasm on the ribosomes themselves, where very simply a small and large subunit will pair together to bind with the messenger RNA. And what will happen is you'll have anticodons which will bind in different types of sites found on a large subunit. Basically you have the peptidyl site, which is the P site right in the middle of the large subunit. That'll be the first one that will bind, and usually that's going to be methionine, which is always going to be the first amino acid to start a polypeptide chain. Very simple, this will start the sequence, and then the next amino acid anticodon pair will actually bind at the A site, and then very simply move from the A site and P site over to the E and P site. Once it goes to the E site, the anticodon will actually be released, and then the protein will start the amino acid sequence. Here we see the full process of transcription for protein synthesis. As I mentioned before, the first amino acid is always methionine. This is going to come from initiator transfer RNA. The anticodon will base pair with the messenger RNA. The anticodons will actually match up with the actual DNA strip that it was translated from. This will bind at the P site of the large subunit, so methionine will start the polypeptide chain. Very simply, it leaves the A site open, which allows the next anticodon or next transfer RNA to bind with that particular site. Next step is the two amino acids will form a peptide bond, while at the P and A site for the two anticodons for the initiator, as well as the next anticodon down the chain. Once that new peptide bond is formed, the two anticodons will shift to the E and the P site respectively, which leaves the A site open, and that particular anticodon or the transfer RNA will disperse away from the large subunit. And this chain will actually continue until you hit the stop codon to form the last link in the amino acid chain. This can happen roughly about 15 times per second, and actually you can have multiple ribosomes form on the messenger RNA, so you can produce multiple copies of the same protein over and over again. Next we'll talk about cell division, which is the process by which cells will reproduce. So when a cell is not mitotically dividing, it is known to be in interphase. When it is mitotically dividing, it goes through four different stages of mitosis. But to start that off, very simply, it goes through different phases, such as the G phase in particular. In this case, once certain cells are fully mature, some will never leave the interphase. In this case, they go into what's called G0 phase, where it exits from the cell cycle in particular. So neurons, once they're fully mature, as well as some muscle cells, will never fully divide again once they're fully mature. However, other cells, including epithelial cells, as well as some other specialized cells, are constantly going to be going through mitosis. In this case, they'll start in what's called the G1 phase. So the first 8 to 10 hours, the cells are still metabolically active. They start to duplicate all the organelles and components, and the centrosomes that we talked about earlier in the chapter start to replicate in particular. Next stage is known as S phase. This is where the DNA will be replicated. It takes roughly about 8 hours depending on the cell. Next is going to be G2 phase, where cell growth starts to continue, enzymes and other proteins are synthesized, and then centrosome replication is complete. This takes about 4 to 6 hours. And then finally it goes into the mitotic phase, which will get into individual phases, starting with prophase, metaphase, anaphase, and telophase. Each phase itself has its own characteristics as a way to look and see how to differentiate one from the other, as if you're looking under a microscope. So again, interphase is going to be comprised of, in some cases, may only be the G0 phase and never reach any other phase beyond that. But if the cell will be mitotically dividing, they'll move into G1 phase, as mentioned before, then S phase for DNA replication, and then G2 phase will finish cell growth and also finish all the organelle duplication and centrosome duplication as well. So if you're looking under a light microscope, this is a 700 times magnification. We see a cell that's going to be within interphase. These particular slides will be very important, in particular when you take the exams. I will be using these particular slides for test questions. So here we see that the representation of an interphase cell on the right. We see the centrosome, the centrioles, and the paracentriole material. We see they're kind of clumped together in one particular portion just outside the nucleus. We can see a clear nucleus. You can see the nucleolus as well as nuclear envelope. We see chromatin. So the chromosomes have not condensed into the chromatids as of yet. And you can very plainly see a difference between the nucleus, the nucleolus, as well as the cytosol. Now we get into mitotic phase. The first phase that we have is prophase. At this time, we see the chromatin start to condense into chromosomes. This is where we start to see the sister chromatids. We start to see the actual centromere, and we start to see the form arm-like bands, which are going to form the actual chromosome. And at this point, especially in the early phase, we start seeing the centrioles in particular will start to migrate to the poles of the cell to start forming the mitotic spindle, which you may remember is formed of microtubules. This will help kind of pull the cell apart to produce two daughter cells. Once we finish late prophase, we move into the next phase known as metaphase, where very simply the centromeres, all the chromosomes, will line up at the metaphase plate. This is pretty easy to see. You see the centrioles very simply at the polar ends of the particular cell on either ends of the mitotic spindle, and the centromeres will attach to the mitotic spindle to get ready for separation at this point. If you look under the light microscope scan on the right hand side, you can see very easily the chromosomes line up at the metaphase plate, but we do not see separation as of yet. Next we move on to anaphase, where the centromeres of the chromosomes will split, and the sister chromatids will move towards the opposite poles of the cell. When you get into late anaphase, you start to see what's called a cleavage furrow, where very simply we see the pinching off very soon to be of the two daughter cells to be formed from the initial mother cell. So you can see also in the light micrograph there as well that you can see very easily in the early phase you start to see separation of the chromosomes via the centromeres on the right side here, and on the left you see late anaphase where you start to see absolute separation along the mitotic spindle as sister chromatids move to the opposite poles of the cell. And finally you have telophase, where the mitotic spindle starts to dissolve, the chromosomes start to regain their chromatin appearance, and a new nuclear membrane starts to form. You can't really see it too well on the actual light micrograph, but you can see very simply that the chromosomes start to condense a little more thoroughly, and mitotic spindle starts to dissolve, but you do see more of the cleavage furrow up through here, and you start to see the nuclear envelope, at least in some stages, start to reform as the two daughter cells are almost fully formed. So the cleavage furrow will actually pinch off in two daughter cells by a process known as cytokinesis, which is just simply a contraction of the plasma membrane, where eventually each side will fuse together to form two separate daughter cells. And at this point, once we finish up with late telophase, that's when interphase will begin again. Either interphase will continue on to either the G0 or the G1 phase. As it continues in through the interphase, it may go to the S phase and then finally back to G2 phase, where it preps for another mitotic divide. So table 3.3 will give you a quick synopsis of the entire somatic cell cycle as it goes through the different stages of interphase as well as different stages of the mitotic phase. This is a nice little chart to give you a quick synopsis of what goes on during both phases. This is a nice little cheat sheet for you to look at. Very helpful in case we go through any particular questions for the next exam. So control of the cell destiny is all based on the protein cyclin. This will basically turn cyclin-dependent protein kinases on and off to whether we want to start the cycle for DNA replication, mitosis, and cytokinesis, or to not ever continue through this process at all. So depending on the cell, we see three different types of destinies. We see either it remains alive and continues to function without dividing, such as a mature nerve cell or certain muscle cells. It may continue to grow and divide, like epithelial cells that we find in the skin or on the stomach lining, or it could be programmed to die through apoptosis, which we had talked about before with the mitochondria. In this way, it's a controlled way of destroying the cell, but also allowing lysosomes to come in and clean the area up and repurpose the leftover material to use for other cell functions, for neighboring cells, or to be expelled into extracellular fluid. Now this is different from necrosis. Necrosis is when a cell may die either due to lack of resources or damage, whereas there's not enough oxygen, not enough glucose, or let's say it gets burned, or other types of destruction. Unfortunately, this causes more of an inflammatory response, which can be very detrimental to the localized area. We'll talk about it a little more so when we get into tissues, and particularly talk about different types of injuries and how the body responds to them. Next, we'll look at meiosis, which is going to be for reproductive cell division. The main difference with meiosis versus mitosis will give us two daughter cells, each with its own set of 46 chromosomes, so it's a fully mature human cell. For meiosis, our goal is to form from one parent cell to have 4 haploid, 23 chromosome cells per daughter cell. When we talk about mitosis, we're going to have a diploid cell, which is going to have a full set of 23 chromosomes with a pair each. But for meiosis, our goal is to have 4 daughter cells that are haploid with only one set of 23 chromosomes. There's no pairing there. So a lot of processes will look very similarly, except the first round for meiosis is going to look a little bit different than mitosis, whereas the second round of meiosis will look very familiar to mitosis that we just looked at a few slides back. So very simply, a lot of functions remain the same. We look at prophase 1. The centrioles will start to migrate to either side of the pole, but instead of each chromosome lining up at the metaphase plate, you're going to see the chromosome pairs will actually line up at the metaphase plate. And at this point, at prophase 1, before they line up, you'll have pairing over, or crossing over, excuse me, of the non-sister chromatids to allow genetic recombination. This gives us as much diversity in our reproductive cells as much as possible. Then very simply, once we get into metaphase 1, you see that centrioles will have fully migrated to each side of the poles. All the chromosomes are at the metaphase plate as pairs of chromosomes instead of every chromosome lining up at the metaphase plate that we find in mitosis. Once we get out of metaphase and going to anaphase, you start to see the chromosomes start to be pulled apart towards the poles, and in some cases you may see the beginning of the cleavage furrow. Finally, it'll reach telophase 1, where you start to see a reformation of the nuclear envelope. Very simply, you'll see the centrioles pull the two daughter cells apart, and the cleavage furrow will continue through cytokinesis and pinch off to getting a full half of the cytoplasm from each of the daughters to each of the daughter cells. Once we finish the first round of meiosis, we move to meiosis 2. Meiosis 2 and mitosis will look almost exactly the same here. So we'll go through the same phases here. So once we finish meiosis 1 and telophase 1, we go directly into prophase 2 in meiosis 2. So again, we have the fully formed nuclear envelope. We see the centrioles have not migrated yet, but they're on the process of migrating to the poles. But the nuclear envelope is still fully formed at this point. When you get into metaphase 2, the nuclear envelope has dissipated. We see the centrioles have moved to the opposing poles. The mitotic spindle has been formed, and your chromosomes will line up at the metaphase plate. Once we get to anaphase 2, we start to see that the chromosomes are starting to be pulled apart towards the opposing poles. You start to see, in some cases, the start of the cleavage furrow. And finally, we finish off in telophase 2, again, very similar to mitosis, where again, the cleavage furrow will continue to pinch off the remaining bits of the plasma membrane. So we have 2 equal amounts of organelles and the cytosol. And at this point, you start to see the nuclear envelope start to form, and then you're left with 4 haploid daughter cells from the one parent cell. So cell diversity, we'll talk more about this when we get into lab 3. In fact, we'll have something to do in lab in particular. You'll be creating 3-dimensional models using either Play-Doh or clay. Just looking at different types of cells found throughout the body. We'll introduce you to a few of them as to why they hold their shape, gives a direct relation to what their function may be. So for example, if you look at the sperm cell, we see it has a flagella with a very small head. As mentioned before, it is sparse as far as organelles go. It does have quite a bit of mitochondria and also lysosomes at the head of the sperm to penetrate the oocyte. Naturally looks dramatically different from the smooth muscle cell to the upper right-hand corner here. That has contractile tissue to allow contraction usually along the intestinal tract or along the cardiovascular areas. So it allows contraction and relaxation. You see an epithelial cell there with microvilli. So the microvilli there is more to increase the surface area for maximum absorption of different types of products. You see the red blood cell, which is a biconcave disc. As you can tell, there is no nucleus there. This is primarily for transporting oxygen and carbon dioxide throughout the blood. And finally, you see the nerve cell, which shows the dendrites, which are the stalk-like projections off of the cell body or the soma, and a long axon that travels down to allow the action potential to travel from the soma down through the end plate of the axon. So we'll talk a little more about this here. For those of you, again, in lab, we'll be talking about what kind of cell that you want to construct. Those of you who are doing online will be doing something a little bit different. I will give you guys continuing information on that as it comes. Now, as we talk about aging in our cells, as we know, obviously, certain types of processes, as we age, start to either slow down or cease to function altogether. So as we can see, cells will gradually start to deteriorate in their ability to function normally. A lot of times they kind of respond to environmental stresses. We do see certain things start to slow down as quickly as age 20 to 25. Certain things you were able to do when you were younger, all of a sudden your recovery is a little bit weaker than it was before, and as we age and get into the 30s, 40s, and 50s, as well into the 60s, we start to see other phenomena occur, which we'll talk about as we get into each particular tissue chapter. Our number of our body cells start to decrease, so naturally with the lesser number of body cells available, then the ability to mitopically divide also decreases as well. So if we don't have enough body cells to take care of certain processes, we also lose the integrity of the extracellular fluids or the extracellular components of certain tissues. In this case, which comes no surprise to many of you, cartilage, blood, muscles start to lose their ability to repair and bounce back from certain types of injuries. This could be from free radicals or oxidative damage, Most of the patients that I deal with, we do talk about taking antioxidants in particular to inhibit the formation of free radicals, so this way cells can function as long as possible. So most oftentimes we'll recommend people take zinc, selenium, vitamin C and E, try and have them stay away from fatty foods, fried foods, and so on and so forth, as these can definitely speed up the aging process.